morning, everyone. I guess you've heard this a lot in the past 2 days, but it's great to be back at a meeting in person. I've been missing this. So today I'm going to talk about some work that has been happening over the past years and is still ongoing. And I want to start with the acknowledgements. So this large part of what I'm going to talk about is done together with Matteo Tomasini, who was a PhD student at the University of Bern and is now a postdoc at the University of Gothenburg in Sweden. And it's essentially based on two papers published and one preprint that you can find here. So I'm going to talk about evolutionary rescue, and just very briefly to make sure we're all on the same page here. So this is an idea that has been mathematically formally introduced in the 90s by Rich Gomarkiewicz and Bob Holt. And the idea is simply that, so this plot here shows, do we have something to point? No. Okay, so essentially you have on this x-axis some, let's say, population of bacteria or any kind of population. So you have the population size on the x-axis of this red line. And then some environmental change happens, so some stress. This could be anything from climate change or if just applying antibiotics to a population of bacteria. And what will happen, let's stick with this example of antibiotics and bacteria, that the population size will decline. So we see this in this first phase, population size will decline. And, well, the goal of applying antibiotics is to kill bacteria. So, but what can happen is that a new mutation appears that rescues the population from extinction. So this would be this blue line here, and this then leads to this transition phase where we have a switch in the population dynamics and then an increase again. So this has been studied under quite a couple of different names in medicine, agriculture, conservation biology, and yes. And I think this is one of the examples of evolutionary theory that has been, or where key predictions have been confirmed in laboratory conditions extremely well. So a few things that have been found is that the most important factors for evolutionary rescue are clearly the population size. So if you have many individuals, you have more chances to get these rescue mutations. But also if mutation rates are higher, or if the other factor that plays a critical role is how severe the stress is, so how quickly population size declines. And in antibiotic resistance, but not only, so in species live in space, space seems to be an important feature, and this becomes very clear when you look at biofilms. So biofilms are these structures that bacteria often build when they're growing, so they have this layer of mucus, and this can lead to very complex, spatially structured populations. And especially in Pseudomonas, so a very clinical, very important pathogen, so this biofilm is associated with a massive increase in the evolution of resistance. And this is partially due to the fact that this layer that is built around this bacterial colonies provides some buffer from antibiotics, so the antibiotics need to diffuse through this, but also there are genetic components and emergence of these genetic components that leads to resistance seem to be amplified by the presence of biofilms, sometimes up to the likelihood of getting, or the probability of getting rescue is 1,000 times larger than compared to well-mixed populations. And population structure, migration or gene flow, and evolutionary rescue has also been studied experimentally, more from a first principle point of view. So about 10 years ago, Graham Bell and Andy Gonzalez made a very interesting metapopulation study where they had wells filled with yeast, and this yeast population that was highly structured was exposed to a salinity gradient that would essentially kill these yeast populations. And then they implemented, using some robot, migration between these wells. And they tried either what they called global migration, which is just taking a sample from each well and then redistribute it evenly, or just allow migration between nearest neighbors, which would be the island as a stepping stone model, well, in different terms. And from the modeling side, there have been a lot of models about spatial structure and evolutionary rescue, but one I want to highlight is a study led by Hildegard Wicker from 2013, where we did a detailed mathematical analysis of a structured population, an island model. So where you have these migrant pool, and so you take a couple of migrants and redistribute it over the whole population. So this would correspond to the global migration scheme in Bell and Gonzalez and Bell experiment. And both of these studies find that gene flow can be beneficial for evolutionary rescue. So here, this plot, both plots show the probability or the number of rescue events as a function, in this case, of the migration rate, and we see this peak for intermediate migration rates. And here, it's just different speeds of the environmental change versus the occurrence of rescue events. And the leftmost bar are isolated population without gene flow, so the darkest one, and the others are different forms of migration. So, and Wicker et al. Used this island model, which is technically not spatial structure, it's more population structure. And they could find, in some cases, very nice analytical results, but mostly they had to evaluate some integrals numerically to find solutions, and the other cases are based on simulations. So what we then try to do is make a simple model of space and then try to see if we can find some conditions for when, under which conditions do we expect gene flow to actually be beneficial. So, and we choose a model like this, where we had two demes, two subpopulations that are connected by migration. So we start with a two-deme model and then go to multiple demes. And then we have this gradual deterioration. So at time zero, before time zero, both demes are in the original environment. Then we have a change where we have original and deteriorated, and gene flow between them, and then both are deteriorated. So in the original environment, these rescue mutations have lower fitness than the wild type. So we take the wild type has a fitness of 1, so that's the average number of offspring they have. And here we just reduce this by number s. And the rescue mutation in the deteriorated habitat, so this would be the one that say red is the presence of antibiotics, has an effect set that is larger than 0. So 1 plus z is the fitness. So this means rescue mutations would grow or will lead to positive growth in the deteriorated habitat. We can then write down a formula for the probability of rescue. So that's relatively straightforward probability theory if you want. So here we have just the probability for no rescue, for no successful rescue mutation in a given generation, in a given team. So Ni is the population size, U is the mutation rate. So for a small mutation rate, U times N can be seen as the probability of a rescue mutation occurring in this generation. And then Pi of T is just probability that this mutation actually manages to spread, in this case, stochastic loss early on. And then we can approximate this by switching to an integral here. So this is pretty straightforward and can be found in literature. But the key ingredients here, and that's why I'm showing this, are the population size dynamics over time and the fixation probability of a given mutation as a function of time, and I is the deme in which it occurs. And so our model should look something like this. So on the x-axis with time, on the y-axis with population size, we have the two demes here. So we have first deterioration, so after time 0, we go into phase 1 where we have deterioration in one deme, but not the other, and then both are deteriorated after a time of beta generations. And whenever deme deteriorates, we assume that the population size declines at the rate r. Otherwise, we have some density regulation that keeps it at or close to carrying capacity. This can be modeled under some assumptions with relatively simple differential equations that give us the dynamics of this black or blue lines here, which is the resident population size of the wild type. Of course, before phase 1, we just keep it at carrying capacity, and after phase 2, both of these demes would decline at the rate r. And So population dynamics are relatively straightforward to model in this case. We do it deterministically. And the other thing we need, the other ingredient is the rescue probability. And again, we have two simple cases here. So in the phase, before phase 1, everything is the deteriorate environment. We can just use Haldane's classic formula from 1927 to say, well, in the branching, if you model this with a branching process, the rescue or the probability of establishment is 0, because if you're selected against, so originally rescue mutations are selected against, well, there's, in a very large population, there's no chance for establishment. After phase 1, where both teams are deteriorated, well, we're again in a uniform environment, so we can use this 2Z formula. So Z is the selective advantage of the rescue mutation, since it's just called this classical branching process result. Gets more interesting in this phase, so where we have a typical local adaptation scenario, so we have a mutation that's good in the red environment, but bad in the green environment. So, and so we can model this by a two-type branching process, assuming that the types refer to the deme in which the wild type, the mutation is currently residing. So this branching process approximation allows us to ignore essentially the resident population dynamics, and because we assume that the number of offspring of individuals are independent of each other. We can then write down this matrix of the average number of offspring of these two types. So it's relatively straightforward. We have the fitness of a type. So type 1 has fitness 1 plus Z, so you produce 1 plus Z offspring. Then 1 minus m half, where m is the migration rate, stay in this team, and the other m half migrate to the other team. And the same for type 2, so it's a fitness times the migration rate, or the probability not to migrate. And then you can use this if you assume that this is a slightly supercritical branching process, which essentially just means that Z is not too large. More technically speaking, we need to look at the eigenvectors of this, eigenvalues of this transition matrix, show that the leading eigenvalue is 1 plus a small number. And then we can essentially calculate the establishment probabilities of such mutations. So where 1 or 2 refers to the deme, and we can find that this is more or less an average over the values Z and S. So this is the selective advantage of the mutation. This is the selective disadvantage in the other deme of the mutation. And then we have these weights that depend on the migration rate or the selection differential relative to the total strength of migration and selection together. So these formulas look like this, and are very good approximations if you compare it to simulations. So we have different lines here. So from these lines down here to the black line here, we have increasing selection against the rescue mutation. So we have the migration rate here and the establishment probability here. If the mutation occurs in the deme where it's adapted at, and in the this is when it occurs first in a deme where it's not adapted at. And we see that, well, this is a function that declines with migration rate, and this is a function that has an intermediate maximum. Maximum for intermediate migration rate, which makes sense because if you're in the wrong deme, you need to migrate into the right deme so that you can then spread. And what we can look at, we can also look at what is if we average over these two cases, because the rate of local adaptation, if you want to call it, is like the population size in deme 1 times the establishment probability times the mutation rate plus the same for this. So what's the overall rate at which locally adapted mutations become established? And this would be given by this, where we just average these two establishment probabilities over the relative sizes of teams. So it would be proportional to this number. An interesting thing that we can see is that gene flow increases this rate of local adaptation, at least for small migration rates, if either N2, so this is the population size in the deme where the mutation is not adapted to, times the selection coefficient of the mutation in deme 1 is larger than the same, like the population size in deme 1, so this is the one where the mutation is adapted to, times the negative effect of being there. I'll go quickly show this in a figure to explain what this means. Same result as if we assume asymmetric migration. So what this means is essentially in any situation where we have a small population size in the deme where we need the rescue mutation, and a large population size in the deme that in our source that provides individuals that migrate into this deme, and migration will be beneficial. And the same holds if we assume asymmetric migration rates instead of asymmetric population sizes. This also means that this scenario where we have, so this should give us some intuition about when do we expect that gene flow should be beneficial for evolutionary rescue in such a scenario. Namely, when this phase one is relatively long and relatively pronounced. So that means we have a long time where we have a small population size in the deteriorated environment and a large population size in the original environment. Right, so this is what this formula says. So we can check this by just plugging now our population size dynamics and our establishment probabilities in this formula. And this is what we get. So this is just a variety of parameter combinations. The lines are the analytical predictions and the points are simulations. So we have the probability of rescue on the y-axis here, the migration rate on the x-axis, and from down here down to up here, so along this second y-axis, we see that rescue mutations that become increasingly more costly. So they have a fitness disadvantage in the original environment. And here we have an increase in either how severe the environmental changes, like how quickly population size declines, or how slowly the change happens across space. So these seem to be two important parameters. You will see in a moment why I chose this. And we see here up here the rescue probability is low and almost straight line. And up here in this corner, we see that we have quite a nice increase of the rescue probability of migration rate. And yes, so the only range where the approximation doesn't work quite well is here. And that's simply because this is where the establishment probability hits 0. And in reality, if you have small population sizes, you still have a small chance for a mutation to get established, even if it has actually slightly deleterious for some time. And therefore, we don't get this smoothness in our curves. Okay, but since we're mostly interested in this initial increase of, so when is some migration actually helpful, we don't care so much about this. So what we can then do is ask again, when is some migration beneficial relative to no migration? So we just take the derivative of this theoretical formula with respect to the migration rate, evaluate it at m equals 0, and ask when it's larger than 0. And if we ignore standard variation, so any mutations that are present at the beginning, then we find this simple condition. So the relative cost, S is the cost of the rescue mutation, Z is the advantage of the rescue mutation. If this relative cost of the rescue mutation is smaller than how quickly the population size declines times how quickly the change of the environment occurs across populations. So if theta is large, it happens more slowly. If R is large, the population size decline is quickly. That's exactly when we would predict that gene flow is beneficial for evolutionary rescue. And when you look at this, so the blue ones are the ones where this condition is not met. The red ones are the ones where this condition is met. And I mean, it's a rule of thumb, right? There are some approximative steps in here, but by and large, I would say that it predicts this behavior relatively well. Okay. This is just a small note, so if somebody's interested in this, you can look at our recent paper. So we can also extend this to include standing genetic variation, which isn't really changing much. It just dilutes the advantage of gene flow a little bit. We can also include asymmetric gene flow or carrying capacities. So essentially, we then have to have this F index, which gives us the degree of asymmetry in the model and modifies our equation a little bit. But before I go to the next step, I just want to quickly wrap this up, that we found this simple condition where gene flow facilitates evolution rescue in this two-deme model if environmental change is slowly occurring across these two demes, but it's very severe within each deme. So these are the ideal conditions, and if rescue mutations are not too costly relative to their advantage. So if the system shows source strength dynamics, where we have one population providing lots of influx into the team, then we expect that this gene flow should be beneficial. And now, very briefly, let's see if we can extend these results to multi-deme models. So we have the island model, where we have gene flow between any pairs of teams at the same rate. And another extreme would be the stepping stone model, so a model of a spatial model where we have a one-dimensional array of teams that are connected with migration. And so this is under review, hopefully out soon. But you can find the preview in the bioarchive. And the results are not overwhelming in a sense, because we see qualitatively very similar behavior across most parameter ranges. So here's a comparison between the stepping stone and the island model. Stepping stone is in gray, island in black for 4 or 16 demes. And what we see is that, well, qualitatively these images look very similar, except that we see like a shift in the migration or on the x-axis between these two, or a stretching of the x-axis. And there's a relatively straightforward solution to this, because in an island model, gene flow is only occurring between neighboring teams, so the effective gene flow between the two habitats is very strongly reduced as compared to the island model. And if we very roughly account for this, then these lines become almost overlapping. So this suggests that the most important thing is not the migration rate between teams, but the migration rate between the different habitats. And this, of course, changes over time and over space. But if you can somehow quantify this, even just approximately, you can really, in most cases, not in all, there are some exceptions, but in most cases, you can map the island model and the stepping stone to each other. And so essentially, understanding the island model should give us also a lot of information about the stepping stone model. I think one interesting difference between these two models in the stepping stone and the island model is that if you look at the degree of fragmentation, so essentially we take a population, we keep the total population size constant, and then just distribute it over a certain number of demes. We also keep the total time until the whole environment is deteriorated constant. So that means if you have 16 teams, in each team there's a smaller population size than if you have just 4 teams. And so this is one example of choosing where the pattern is very clear. But so the yellow line is weak migration rate, and the blue line is strong migration rate, and the other values are in between. And in the island model, so this has already been shown by the Hildegard-Oecker and colleagues in 2013, in the island model, the number of teams doesn't really change much, which makes sense, right? It's since migration is going between any pairs of teams anyway, how many teams you have is not that relevant. But in the stepping stone model, for a given migration rate, the effect of the increasing fragmentation can go in both directions. So if we have strong migration, increasing the number of teams, the population structure, can be very beneficial for rescue. If we have weak migration, not much is happening. And if we have intermediate migration rates, we might actually see that this disadvantage gets reduced. Okay, so I'm happy to discuss this more in a break, but I think it's time to wrap this up. So the first part showed that gene flow can facilitate evolutionary rescue if environmental change is severe, but it occurs slowly across space, and if rescue mutations are not too costly in the original habitat. So that means the cost of an antibiotic resistance mutation in the absence of antibiotics. Spatial structure matters to some extent. They show qualitatively very similar behaviors. The process of gene swamping, so not being able to establish a mutation because of too much gene flow, is less important in the stepping stone model. But the effective migration rate between habitats rather than teams is the key quantity here. And the degree of fragmentation is more important in the stepping stone model, because simply the degree of fragmentation determines our effective migration rate. So for the same migration rate between teams, we might have different effective migration rates depending on how structured our population is. And I think, yes, that's a good point to stop. So happy to take any questions.
Is there questions?
Thank you for your talk. Did you investigate, or do you plan to investigate continuous habitats?
I ran out of time. Yes, so and I would be very happy to discuss this here because I know there are lots of people who know this much better. But yes, we look at continuous space to some extent. It's just very hard to get something as nice as in the simpler models, but we have some ingredients figured out. And I also want to look at something where we, instead of migration, look at mutation in the genotype space, like for viral species, which would be a very similar model. You have to just change the diffusion kernel a little bit. Yeah.
Thank you for your talk. I just had a question about clarification. So you had some population size dynamics, but as I understand, there are mutational rare enough that you don't have to, that you don't model the joint effect of mutation and population size, because like your leading eigenvalue was not having any effect of difference in team size.
That's an important point. So we model just the blue lines essentially here, just the resident population size, and the idea is that as soon, I hope I understood you correctly, but the idea is that as soon as you have a number of rescue mutations that is so large, the population, that it actually would change the dynamics of the population, you already have rescue, right? If the mutants start to grow and would interfere with the population dynamics, then yeah, you already have rescue. So that's an approximation, of course, but it works quite well.
Okay. Okay. But if the mutation rate was a bit more frequent, then you will couple the two.
Yes.
Okay.
But usually these mutation rates are not that large. So we used mutation rates up to the order of 10 to the power of -4, which is already very large.
Okay.
And yeah.
Okay, thanks.
Sorry, I'm not sure I understood. When we have migrations, there is a demographic effect. We bring some more susceptible, more hosts in the red environment, and there's gene flow, which is separated, right? And I did not really see how we could distinguish the two effects.
So can you repeat the first effect?
It's a demographic effect. You have a constant inflow of individuals, and so it increases the ones that are susceptible to mutate.
Yes. I mean, yeah, that's a good point. And that's, I mean, these are, when you see this, why is intermediate migration optimal and not large migration, right? So the demographic effect is increasing with increasing migration rate. But so that's one effect. And so that's the demographic rescue effect, but the like genetic rescue effect decreases with increasing migration rate at some point, right? So it's, if you take the black line here, it gets 0. So even if you have a very strong demographic rescue effect, a rescue mutation cannot establish, and eventually the whole population will die. So these are these two effects of gene flow: the demographic rescue effect and the genetic rescue effect. And we model them really separately. We assume we can model the demography and we can model the fate of the rescue mutations independently, and then just plug it together. Yeah.
So you mentioned the experiment of Gonzalez.
Yes.
It's the assumption that the deterioration moves slowly across space. Is it also in the experiment, or?
They used several. Yes, they had you see this here. This is reflectedness on the leading edge. You have a different environment than at the core, and then you had like one constant change, a slow change across space, and a fast change. So and I think that's the gradual change across environment is a key ingredient for this process to be important. Yeah.